All right, we're going to go ahead and get started. Go ahead and take your seats. Have you guys been enjoying the day so far? All right. We're almost there, home stretch, guys, okay? I'm delighted and very honored to introduce our next speaker. Dr. Dermot McGovern is a professor of medicine and director of precision health here at Cedars-Sinai. He completed his PhD and fellowship at the University of Oxford in the UK. Besides being a world-renowned geneticist, he is a member of the steering committee for the International and North American IBD Genetics Consortia, as well as the Crohn's and Colitis Foundation Microbiome Initiative. And in addition, he is my esteemed co-chair here for the Los Angeles Crohn's and Colitis Foundation Medical Advisory Chapter Committee. Okay, we'd like to go ahead and introduce him to share on updates in inflammatory bowel disease research. Please give him a warm welcome.
So good afternoon, and it's you I should be applauding, the guys who've made it to the end. Congratulations. So when we sort of put the program together, obviously it was our intention to put the hard science right at the end when you were most awake and with us. So I'm not going to be asking any questions. There's no quiz or anything at the end. And I really just want you to walk away from this with some of the ideas and the concepts, not really about the details of where science is going. And of course, in about half an hour, I can't cover everything in the science. So I'm going to try and cover things which I think are going to have the most impact on the clinical side over the next few years. So, I said there was no quiz. There is one. I lied. Right at the beginning. So, does anyone know where this quote comes from? No. I wish I'd been that intelligent to say that. Okay. I've asked about 10 audiences and not one single person's ever got this right, so you're fine, okay? This actually comes from War and Peace by Tolstoy. And I think this is a really interesting quote because it really summarizes what we're thinking about IBD now, which basically is that if all of you with IBD, either Crohn's disease or ulcerative colitis, or you have family members with that, sort of compared notes, you'd realize that there's a lot of variation within what we call Crohn's disease or ulcerative colitis. And so one of the ways we're thinking about this is we need to break these diseases down into smaller parts to lead to real breakthroughs. And I'm going to give you some examples of that as we go forwards. So if we think about how we thought about medicine in the past, it's really, this is the way. So we really have not thought about pre-disease, or even in people who've just developed disease but are asymptomatic, except for some quite well-known examples of screening. Once people have disease, we think about standard tests, and then we give you a treatment, and basically you know the story. Here's a treatment, try it for 3 months, come back. I know that 1/3 of you are going to respond, but I can't tell you whether you fall into that 1/3 or 2/3. And then you come back and you say treatment doesn't work. So if it doesn't work, we alter treatment. That's not very good. That's not very satisfactory. I think we all think that we would want to do better than that. And I think in the future, what's going to happen is we're going to be very interested in identifying people who are at risk of disease and early detection, because that's where we may be able to intervene with our less drastic treatments. So everyone's interested in the microbiome, but once you've started developing disease, it may be too late for a lot of people before to actually intervene with the microbiome. We already maybe started the whole process going. So maybe microbiome therapies may be very effective in people who are at high risk of disease, who have got very early disease even before they know they've got disease. Once people have disease, instead of just thinking about this as Crohn's disease and ulcerative colitis, we're going to really try and break this down into these subsets of disease so we can get the right therapy to the right patient based on a whole load of molecular sort of markers. And then once we've got people on treatment, we're not going to wait 6 months to say, well, you're going to respond or not. We're going to have a lot of markers that we can use with what we call near-patient technologies. So you may be able to take a finger prick of blood, send it through the mail, and we'll be able to run a whole host of markers on that to actually see whether you're responding to the therapy or not. So much more sort of near-patient monitoring of disease, which I think is going to be very, very important. Of course, we're going to have to check that this really leads to major changes in disease and actually prove that it saves money, because we all know the issues related to the cost of healthcare. So in summary, not for the whole talk, what we really want to do is get right therapy to the right patient at the right time and at the right dose. Very, very important, and we're not very good at doing that at the moment. We've got a lot of work to do. So genetics is really going to help us with this. It's only one part, but I'm just going to talk about it to start with. And you can see here, IBD has enjoyed unparalleled success in terms of gene discovery. That's absolutely true, unparalleled. There's no disease that has been more successful in terms of us being able to identify genes for that. And there are a number of reasons for that, but one of the really important reasons is that the research community is very collegiate. They work together, share data, and that's helped us. And Cedars has been a significant part of this. Has anyone here actually, if you don't have to answer, but has anyone here donated samples to research at Cedars? Okay. So every single one of you who's done that, give yourselves a round of applause, because you've contributed to identifying all of these genes. Because without the patients giving the samples, we wouldn't make that progress. So that's a plug for getting people to help with research, okay? But we've been able to share your data with other people in an anonymous way, which has allowed us to identify these genes. Now, identifying these genes, we've discovered some interesting things. So if we think about the about 200 genes we have for IBD now, what we've been able to show is that the majority, these are the ones in purple dots here, are shared between Crohn's disease and ulcerative colitis. And that's why we see when we look at families where there's a history of Crohn's disease, there's also a history of ulcerative colitis, and the other way around. There are some which are Crohn's disease specific and some which are ulcerative colitis specific, but the majority are shared. We've also shown that there's overlap with other conditions, and in fact, there's overlap between IBD genes and a lot of the other, what people call autoimmune or immune-mediated diseases. So things like spondyloarthritis, other types of arthritis, type 1 diabetes, psoriasis, those sorts of things. So that's why people with IBD are at increased risk of getting these or having a history of that in their family. We did find some overlap with some infections as well, and like leprosy and mycobacterial disease, and I'm going to come back to that in a little while. The majority of this progress was made in populations of what we call Northern Europeans. That's essentially white populations. But more recently, and this is a big interest of ours here at Cedars, we've made some progress in identifying some of these genetics in other populations, so African Americans and East Asians and so on, and that's very, very important for us to do. We also have an interest in Ashkenazi Jewish genetics, and one of the things that we've known for a long time is that there's an increased prevalence of this disease, particularly Crohn's disease, in the Ashkenazi Jewish population. And what we've been able to show, and this just shows it here, is that it doesn't matter whether you have disease or not. If you're Ashkenazi Jewish, you carry more of the genetic mutations that predispose you to Crohn's disease. And one of the reasons that this may be the case is that these genes may have protected populations in the past from some infectious diseases. So here we see these overlap with leprosy and mycobacterial disease. Mycobacterial disease is tuberculosis is one of them. And we know that the Ashkenazi Jewish population had lower rates of tuberculosis historically than other populations that they were living around. And one of the reasons is they may have more of these genes that protect them from that. One of the consequences now is that they have a higher rate of Crohn's disease in that population. So that's a very interesting sort of Darwinian thing where there's been associations there. And this actually is a comparison of the genes that we see in the Ashkenazi Jewish population compared to the European non-Jewish population. And the genes that are associated with the Ashkenazi Jewish population are more associated with response to bacteria that get inside cells, and mycobacterial tuberculosis is one example of that. Whereas some of the genes that we're seeing in the non-Jewish population may be more related to response to fungi. Really importantly, I'm not saying fungi and mycobacteria cause these diseases. What I'm saying is that these genes exist in our population because at some point in the past they may have helped us deal with these infections. So it's a very, very important difference there. So what has the genetics told us? So anyone who's seen me talk in this forum before will recognize this picture. This is the London Underground. And it helps us talk about something called genetic heterogeneity. And essentially, the idea here is that if your destination is Crohn's disease, so you're on the London Underground, you want to go to Crohn's disease, you may not want to go to Crohn's disease, but say you want to go to Crohn's disease, then there are different ways that you can get there, and on the London Underground, those are the different lines, but in genetics, it's different pathways where the genetic mutations may occur. And you can see that there are lots of different pathways. So if you're thinking about that, why do we think that anyone or everyone with Crohn's disease would respond to therapies in the same way? Because they've got there by very different destinations. And that goes back to that first quote that I was talking about. So when I'm sitting in my clinic, I think about the things that research can help me about and can help the patient that I'm seeing. And these are the things that we talk about, and I'm just going to go through a couple of these. We were just talking about 23andMe, and I'm interested to know how many people here have had their genetics looked at. Well, that's pretty good. You're all clustered over there. Do you know each other? That's higher than if I was giving this talk to a medical audience, actually, which is quite surprising. So 23andMe and other companies like that do this direct-to-consumer testing. And so basically the idea of this, if you haven't done it, is that you essentially spit into a little pot, you send the little pot off to the company, they run your genetics, and they see how many of these markers that you carry for disease, and then they send the results to you. This is you sitting in front of your computer, and for those of you who are close to the front, you can see it says here the risk is 15%. Okay, it can tell you whether your risk is 15%. That's the implication. Okay, actually the small print says it can tell you lifetime odds of getting Crohn's disease between 0% and 1% based on genotype. That's not a very good diagnostic test. Okay, this is written by marketing. This is written by the lawyers. Okay, this is very, very different. And actually, in one study, when they looked at 5 different tests for Crohn's disease in 2 individuals, they got opposite results in 3 out of the 5 individuals for the tests. So it wasn't very, very good. And the current head of the NIH says this is sort of the modern version of genetic snake oil. I'm going to show you why. So I know some of you are taking pictures. That's fine. Just please don't take pictures of the next 2 slides. Do we have a deal? Okay. I'll find you if you did. Okay. So this is my data. This is why I don't want you to take it. So I've run the genetics on 23andMe. And it shows what are things I have an elevated risk of. and I actually have a small elevated risk of ulcerative colitis. These are the highest things. But actually my risk is 1.3%, whereas just if you dragged me off the street, what would be my risk? It would be 0.8%. So it's nearly twice as high, but the absolute risk is pretty small. Okay, and this is why this is not a good diagnostic test. I'm at decreased risk of a number of things. Crohn's disease, so my risk is 0.4%, but my average risk would be 0.5%, so really it's not telling me a lot. I can't use this as a diagnostic test like this. You can find other things, I won't go into that. You can find other things about the genetics, okay? So this is my genetics, and you can see that I am the whitest guy there can be, seriously. So my family's lived all over the world, going back generations, no scandal. It's so boring. There is a little hope for me working at Cedars-Sinai, because I am 0.1% Ashkenazi Jewish, and I do have above average Neanderthal DNA. Now let's just go back a second, okay? These are the things that I'm at decreased risk of. So if we're thinking this is going to help us in terms of how we behave, look at the thing I'm at most decreased risk of, malignant melanoma. But we just saw that I'm like the whitest guy there can be. So we've got to be very careful how we interpret this data, because does this mean I shouldn't be using sunblock? No. No, I should not be having any false sense of security with this sort of data. So we have to be very, very careful. Just touching on the Neanderthal DNA, I know you laughed, but Neanderthal DNA actually may raise the risk for some modern human diseases because it actually it survived again through the Neanderthal era by protecting people from infection, and now we're paying the cost for that. So we do have the IBD genome, as we're calling it now. So how can we use this data? So we're beginning to think about this. I've told you that if you take those individual genetic variants, which really is what the 23andMe thing I just showed you, it really can't help us that much from a clinical point of view. But we can think about this in a different way. So what this actually shows here is the genes associated with ulcerative colitis and Crohn's disease. And again, for those of you near the front, you can see that this is something called IL-23R. This is one of the genes we're very interested in. And it's associated with both ulcerative colitis here, that's marked there by that bracket, and Crohn's disease here by that bracket. But you'll notice that it has a much bigger effect in Crohn's disease than it does in ulcerative colitis. So what we can start to do is think about the effect size of these genes. Can we start to use them in more complicated ways? And actually what we can start to see is that if you take every single individual, you can create a Crohn's disease risk score for them by combining all of the genetics. And these individuals here in green have Crohn's disease. These individuals in ulcerative colitis, in this purple have ulcerative colitis. And you can see there's a difference between that, but they overlap. And then when we looked at these scores, we actually found that the scores were very, very much associated. Here are the scores on where you had the disease location, where you had your IBD, going from ulcerative colitis, colonic Crohn's disease here, small bowel and colonic Crohn's disease, and then small bowel disease. So again, starting to help us break these down a little bit here by disease location. We're also interested in disease behavior. Those of you who have had perianal Crohn's disease will know perhaps that our therapies are not very effective in that area, and a lot of people require surgery. So this is an area where we've been trying to work very, very hard at identifying genetic changes. And we published this a couple of years ago now, and while we found no individual genetic variant was strong enough to really help us, when we combined all of the data, we found that a particular pathway had a lot of genetic variants in it that were associated with developing perianal fistula. And this was something called the JAK-STAT signaling pathway. And why that may be important is that we now have drugs, and this was published earlier this year, which target specifically the JAK-STAT signaling pathway. So we would say to people who are developing drugs like this, you really need to try this in our perianal fistulizing disease group, because we genetically think they're a group who will respond. Of course, this is the ulcerative colitis. They also did a study in Crohn's disease. The one group of subjects that they excluded from their study was people with perianal fistulizing disease. Okay, so we definitely need some education. I love telling this story. This is Burrell Crohn and his colleagues. When they described Crohn's disease back in 1932, they described a disease of the terminal ileum, that's the end of the small bowel, affecting mainly young adults, characterized by some inflammation. When a British surgeon described colon disease, so large bowel disease, which looked like Crohn's disease, he wrote to Burrell Crohn, and Burrell Crohn wrote back and said, this is not the same disease. So what we now call colonic Crohn's disease, Burrell Crohn didn't recognize as colonic Crohn's disease. And it now seems that we have some genetic differences between these two, and so Burrell Crohn was way ahead of his time, thinking that we've got to break these diseases down into smaller parts. And this is our IBD population, and a very, very mixed bunch in terms of how the disease is behaving. And what we need to do is think about this, is breaking them down into these smaller groups to understand disease better. So moving on to prognosis, can we predict how people's disease is going to behave? This is a very nice study from the UK from a lot of people who are my former colleagues when I worked there, where they tried to identify genetic variants associated with severe disease. And they did. They found 4 genetic variants that predicted severe disease. And the interesting thing is that, again, like we saw with disease susceptibility, there's associations with infection. So this gene, FOXO3, is associated with more severe Crohn's disease, but the change in the gene that's associated with more severe Crohn's disease protects you from developing malaria and tuberculosis. So again, we're seeing this opposite association, which is very, very interesting. This is a study that was funded by the Crohn's and Colitis Foundation. So any of you who have ever been a member of that, donated money, by definition coming to this meeting, have supported this study. And this is a study that's published this year in The Lancet, one of our most esteemed journals. And basically it was what we call a pediatric inception cohort. So they took a group of kids who'd just been diagnosed with Crohn's disease, and they followed them up to see who developed the more severe disease. And what they were able to show is some interesting things associated with more severe disease. And some of them were the disease location, and some were some serological markers. Interesting, African Americans were more likely to develop one of the more severe forms of Crohn's disease. They also showed some genetic signature and also some microbiome changes, and were also able to incorporate those into what we call a disease model. And so here you actually have a model of predicting who's going to get severe disease based on a biopsy, which actually would be very, very helpful for us going forward. So we need to, as we say, validate this. We need to check that it works in other populations, but there's definitely work going on there. They've actually gone on and done some additional work and found a better way of predicting severe disease. And I wanted to just mention this study because it's one thing to have a biopsy to do this, but a group from Cambridge in the UK have actually found a genetic signature from a blood test which predicts severe disease. And again, the really another important thing about this is they actually borrowed this signature from lupus. And so the same signature that predicts severe disease in lupus predicts severe disease in Crohn's disease. So again, borrowing things from other areas in medicine. We have a lot of existing therapies that are effective, but are associated with side effects, as many of you will know. And one of the drugs that we use a lot are the class of drugs of the thiopurines. So that's azathioprine or 6-mercaptopurine. And I'm sure there's some of you in the audience who've been on those drugs. Now, this is an important study which was performed in a Korean group, and we were part of the collaboration for this, where the Korean group, based in Seoul, noticed that Koreans were more prone to this side effect of bone marrow toxicity, which can be life-threatening with this drug, but actually carried fewer mutations of the one gene that we really knew that predisposed people to this. So they did a study and found another gene, this gene here, NUDT15, and they did some beautiful work showing that the more copies of the genetic change that you carried, the lower the dose of azathioprine you were able to have and the more severe bone marrow toxicity that you had. And the important thing was about this study, apart from this is going to really help us in the East Asian population make these drugs safer, is that this genetic change is very rare in Europeans. But when we looked in the European population, if you carried this mutation, you got this side effect. And we would never have found that without having started in the East Asian population. So it's very, very important for us to learn across results from different ethnicities. We've also been able to identify genetic variants associated with another life-threatening adverse event from this drug, which is pancreatitis. And so now you can see how we can start to build a panel of genetic tests that we would use that would help us make these drugs, using these drugs, much safer. We're interested in whether we can predict whether people, not just from a safety point of view, whether they can respond or not to drugs. And this is a study that we published earlier. The one point I want to make about this study is that when you look at all the different genetics, you can create what's called a gene network of how these genes interact. And you look for things that are what we call the nodes, right at the center of all the interactions. And the one I've highlighted here is something called RAC1. And why that's important is that there are therapies already out there which are targeting RAC1 for a number of different medical conditions. So here we think by finding this, that we could use RAC1 inhibitors in patients who don't respond to anti-TNF therapy. And these drugs have already gone through all the the FDA approval and toxicology and all of that sort of thing. So repurposing drugs from one area to another area in medicine is something that we're very, very interested in, because that means we can get drugs, appropriate drugs to patients very quickly. So we're thinking about redefining IBD, but we're not alone. So are most of the people who are working in other immune-mediated diseases. And so you can actually think where you have a genetic signature that identifies a subgroup of your patients with Crohn's disease, and that signature can look much more like some patients with psoriasis, for example, than other patients with Crohn's disease. And therefore you can think about using therapies in both of those subsets of disease. And in a way, you're sort of ignoring the organ that's been damaged, whether it be skin or the gut. It's based on a molecular signature. So this is one of the ways that we're going in terms of thinking about the disease and prognostics, as we say. So I've sort of shown this before, where we're going to divide disease into different ways of thinking about it. We have a number of different therapies that were available at the moment. And from our clinical trial data, you can start to see where some of these things may be effective, and also where we're sort of missing therapies. And then we look at the pipeline and see, are there genetic signatures that we can see which suggest to us that this therapy will be good for that subset of disease? So here I talked about perianal Crohn's disease. We do have some therapies. They're not great. What about those JAK inhibitors that I mentioned? So that falls under there. So this is one of the ways we think about drug development now. And we really need this, because historically we've not been good at identifying drugs. The pipeline's quite good at the moment, But if you look back, we've had so many failures in IBD. And one of the reasons is that it's always been, this is the historical way of discovering drugs. So you identify a target based on who knows what. Sometimes some very, very flaky evidence. Then you spend millions of dollars going through chemical screens, exposing animals to this, and then finally into clinical trials. And we found to our cost that mice are not men and men are not mice, essentially. And so many compounds make sense up to here, and you give mice the compounds and mice get better. And there are thousands of mouse doctors in the research arena who think they've discovered the next big thing for treating disease, but this is a real problem here because mice have different immune systems than humans. So one of the ways we're thinking about it is perhaps you should start with the genetics. Perhaps you start with what's going on in the human, the underlying causes of this. And we're interested in a number of the genes, but here's just one, TNF superfamily 15. This is a very interesting gene. It was discovered very early. This is 2005 as being associated with Crohn's disease. And it's interesting because this, again, shows you the effect size between different populations. This is the European population, and the effect size is this tiny brown bar here. This is the East Asian population, and this is the effect size. So it's the dominant gene for Crohn's disease in the East Asian population. But in the European population, it is associated with disease, and it's associated with more severe disease, particularly stricturing disease. Okay, I'm really sorry. I should have warned you I was going to put that one up straight after lunch, or it was quite soon after lunch. This is a mouse model, having just trashed mouse models, where they've actually created what we call a transgenic form of that, where you've created a genetic change in it which is found in the human. And in human Crohn's disease, we know you get strictures in the gut and other places. You can also get skin lesions as well. This is something called pyoderma gangrenosum. And in the mouse model, you get similar strictures, and you also get similar skin lesions as well. And the really interesting thing in the mouse model is that you can recreate what we call fibrosis. This is the thing that causes strictures, those narrowing. I heard Dr. V talking at lunch about narrowings in the small bowel. The process is fibrosis. Here you can see the fibrosis. You actually cause the fibrosis. You treat it with placebo. The fibrosis continues to get worse. If you actually treat it with a drug against this gene, the fibrosis reduces. You actually can reverse fibrosis. This is one of the holy grails of research in IBD, is to see if we can reverse fibrosis. Because if we reverse fibrosis, we can actually think about putting surgeons out of business. Wouldn't that be great? Those of you who know Dr. Fleschner, don't say anything to him. I'll be in big trouble. Okay, so this is very, very exciting. And the other exciting part of it is that because we started with the genetics, we can actually identify a population who we think will particularly respond to this, because the genetics tells us that that change is important in that population. So when we do a clinical trial, we can ignore parts of the Crohn's disease population and just get people into the clinical trial who we think are going to respond to this drug. And that means we don't expose people to potential side effects who are less likely to respond. It also means that the trials can be smaller and cost less money and get these drugs cheaper and quicker to patients, which is really, really important because that's a big problem for us at the moment. And we're not alone in thinking about this. And this goes back to the repurposing of drugs. This was a really interesting study in rheumatoid arthritis. And what they did is they started with the genetics. They looked at some of the genes, the genetic changes, what we call the polymorphisms. The genes that were affected, were there drugs that were targeting these genes? And they found some that they already used in rheumatoid arthritis. but they also found some others that were in use in other parts of medicine. So again, this is about repurposing drugs from one branch of medicine to another. Every now and then we have some very, very disruptive technology, and the genetics has been one thing where we've been able to start doing the genetics on millions of these genetic variants across any one individual and running those chips very, very quickly. This is another technology which I think is going to really change things that we've done. And basically, that is that you can take blood from people, generate these stem cells, and you can take the stem cells and you can drive them to different organs, including the gut. So this is a gut in a dish, essentially, and it has all the cells that we're interested in. And this is incredibly useful for us for driving our discovery. And people working here at Cedars that we work with at the Regenerative Medicine Institution have gone one step further and are actually using this chip to actually put organs on a chip. And why this is exciting, I think I'll be able to show you. Oh yes, the movie works. Thank you. This is the chip. Okay, it's about the size of a quarter. When you cut through it, what you can actually see is, and we borrowed this from the lung people, so you'll have to forgive us for that. There's a membrane in this part of the chip. It says lung cells, but we put basically the lining of the gut there, and you can put blood vessel cells underneath. Remember, you've started with a blood test with everyone, and you've driven these. It has a vacuum channel on each side, which is really, really important. For the lung, it reproduces breathing. For the gut, it reproduces that peristalsis, because that makes the cells survive. Here it says you can flow air across, but in the gut, we can flow the microbiome, the bacteria that are in the gut across, and then the blood channel underneath. And here, I'm going to show you what that's like. So here you have the bacteria flowing across, and you can see that for some reason, sometimes bacteria stick, and we don't like that. They're close to the immune system, and when that happens, you can start to develop Crohn's disease. But your body responds to that, and you can see these are white cells that come through, go through that membrane, and actually attack the bacteria. So this is white cells flowing past on the chip when nothing's going on. And then you can see when there's an infection, these white cells start to stop and stick and start to do things. Can you see that? There's much more of a population of these white cells here. And this is an example. This is a white cell, and it's trying to find its way from one side of the chip to the other, and it comes through that hole in the membrane and then comes through to the other side. And this is another example of where this white cell is going around eating up the bacteria that are there. Now, this is a very important process. I showed you lung. This is a very important process for fighting infection, but it's one of the things that drives inflammation in the gut. You get this what we call an inappropriate response to bacteria, because your gut is loaded with bacteria, as I'll show you in a second, and most of the time we don't respond to them. Our immune system doesn't respond to them. I just put this in as a backup in case the movies didn't work, but here you can see this is the size of that chip. And in fact, this is a picture of the chip, if I can get this to work. Okay, 2 out of 3 movies worked. But you can see the most important thing. This is a cross-section through what we would see on top of the chip. And you can see these structures here. These are villi. So we're really seeing what we see in the gut in this chip, which is absolutely fascinating. So as we sort of move forwards, we can create IBD on a chip, because not just the gut, but you can actually take cells from you and make your immune system and your microbiome and expose that and sort of recapitulate that. And this allows us to look for what causes IBD, but very important in drug development, and you can actually personalize it to the patient, because you can actually expose that chip to the drugs before you expose them to the patient to see what happens. And that's where we hope we're going with this. And because you can do this for lots of different organs, you can put them in series or parallel. I always used to get confused about that. These chips, where you've got one for the gut, one for the blood-brain barrier, one for the liver, and so on, to make sure that even if things are working in the gut, you're not causing damage in the liver, and so on and so on. So this is incredible technology, which we're going to see a lot about in the future. What about cures? We're very, very interested in cures, of course, and some place where we've had success is when we think about individuals with very early onset IBD, because I told you about what we call oligogenic and polygenic diseases, which is where lots of genes contribute. In very early onset, we're seeing what we call monogenic. That means you have one mutation, you have the disease. There's this 100% relationship. And we've been able to identify cases like this poor child who had severe colitis, fistula, skin inflammation, lots of infections, no wound healing, joint symptoms. And the reason was that this poor kid had basically was the product of two first cousins marrying. They both carried a rare mutation. The poor kid inherited both of those rare mutations and basically had a knockout of this gene. None of our therapies worked, but through some biology, they were able to work out that this was all coming from the bone marrow. The child had a bone marrow transplant, and it cured their Crohn's disease. So you can see how we're breaking chunks of these diseases, and this is sort of the low-hanging fruit, but it's really paving the way. So there are a number of challenges as we try and overcome some of the barriers associated with getting these to the clinic. We need to define the genetic components of disease, and we're really beginning to do that. We need to think about the human microbiome, and I just want to give you a couple of stories about the microbiome from recent years. Do you remember I told you about that CCF-funded pediatric inception cohort? Well, in that, they were able to show some very interesting microbiome changes, some of which we knew about before. But interestingly, they were able to create what they called a dysbiosis index, which is a collection of mycobacterial signatures, and these were associated with more severe disease. So again, thinking about, can you predict the future? Talking of the future, this is a study that we published last year, where we were able to take subjects who had Crohn's disease and look at their family members to see were there individuals in the family who had a microbiome signature similar to the Crohn's disease. And we were able to identify that some of those subjects probably had inflammation in their gut, even though they had no symptoms. And other individuals had a microbiome that probably predisposes them to developing Crohn's disease. And remember when I talked right back at the beginning, we want to identify those people who are at high risk, because maybe we can intervene with the microbiome here before they start to develop disease and so on. And then I just want to tell you about this one other story from the microbiome. We discovered a gene for Crohn's disease, and this gene is called, very catchy name of SLC39A8. What it is it's a zinc transporter. It helps you get zinc into cells. And that was interesting because we knew that zinc deficiency is associated with an increased risk of Crohn's disease. This gene isn't associated with ulcerative colitis, but is associated with metabolic syndromes like hyperlipidemia, obesity, and so on, as well as schizophrenia. And what we were able to show is that if you carry this genetic variant, and you're healthy, it causes a shift in your microbiome, which is in the direction that we see in people with Crohn's disease, but also in a direction that we see with people with obesity. And so it was maybe the microbiome that's driving this association that we see for both Crohn's disease and also these metabolic syndromes as well. And then finally, what we need to do is get practical systems for clinical genomics informatics. And what does that mean? That reflects this, okay? So this is probably where medicine may be going in the future. This is a professor at Stanford who basically decided that he was going to sample himself for a period of about a year and see just basically what happened to all of these different things, proteomics and metabolomics and so on, over a period of a year. And unfortunately for him, he developed type 2 diabetes in that year. The upside of that was that he managed to get a very nice publication in Cell, which is one of our major journals, because he was able to then go back and show from his data that he could predict that he was going to develop type 2 diabetes, which is really, really interesting. But look at this. How can we manage all of this data? This is a real problem for us, and this is reflected here. So, we make clinical decisions by what we call clinical phenotype. So that's when you see your physician, he or she asks you lots of questions, examines you, and so on, and tries to make decisions with you based on that. We also make clinical decisions by sociocultural determinants of health. I've given up trying to guess what that means. It used to be called Obamacare. I no longer know what that means. But it obviously influences our practice. But increasingly, we're going to take all of that data that was on the previous slide and make decisions. Now, the problem for us is, no matter how intelligent we think we are, we're just about down here somewhere. And we're really going to struggle with that. So we've been working on mechanisms that we think may be helpful in the future. And of course, this is a program that's now on a cell phone and an app, okay, where basically you can take a whole load of risk factors that include clinical, genetic, and serological, and you can plot, based on those factors, what your risk of developing severe disease is. And this is someone, if we didn't treat them, what their risk of developing severe disease over a 3-year period is. So you would think if they had that profile, we really want to intervene early with some of the drugs that can stop some of these complications developing. In contrast, this is someone else's risk, which is very, very low, and we would probably not intervene with those drugs right up front with this individual. Of course, you don't stop monitoring and so on, but this helps in terms of thinking about risk. So just to summarize, we really are at the age of the IBD genome, which is giving us new insights into disease pathogenesis. We have a problem, though. We do have opportunities for biomarker and drug development and thinking about more homogeneous groups of patients, but we're not quite there. So I don't want you to run out to your physician tomorrow and say, I heard this talk, and here's my genetic data, you need to make all these decisions about me, and I'm going to get my microbiome done tomorrow, and then you can plug it into that app, and we're good to go. We're not quite there. We still need a lot of work. It's very, very important to make that point. But the pace is incredible in terms of discovery and thinking about translation and so on. And of course, we're not alone in this. We're really trying to think across diseases and learn from each other and so on. So finally, if I can get to the next slide, I just need to acknowledge that I'm very fortunate to work with an amazing group of people, and the work that I presented today is largely their efforts and also some very, very wonderful collaborators, and of course funding, and particularly today the CCF. So thank you very much. Happy to answer any questions.
Again, thank you so much. Let's give a hand to our speaker, who's always wonderful. We're very, very fortunate to have him as a part of our foundation. So just a few quick housekeeping items. Please remember to turn in your blue evaluation forms. You can turn them into the front desk. And again, thank you so much for coming. This is a huge opportunity for all of us to learn so much, and we appreciate all of you being here. Before everyone leaves, I would like to give a shout out to our education manager, Tiffany Jordan. Can you raise your hand? She plans this entire conference working very closely with our CMAC, and we could not do it without her. And I know we joke about it, but Tiffany, you are the wind beneath our wings. Again, thank you so much for coming, and please enjoy the rest of your day.
I don't think so.